So the dominant sequencing technology that is applied throughout the world, probably one of the biggest life science companies out there, based in San Diego, is Illumina sequencing. So Illumina, I think right now, somebody told me something like it's it's harder to get a job at Illumina than is to get into Harvard or something like that. I don't know, but regardless to say, they're a big deal when it comes to sequencing, and they are by far the platform that is used in the most places and is the most widely applied. And one of the reasons why is that their library preparation steps are fairly straightforward, and most people can do them, and their instruments are cost efficient, reliable, probably the most accurate sequencers aside from maybe a Sanger, and they put out a lot of information. They put out a lot of sequence, so they're very when we talk about massive parallel sequencing, these guys are definitely it. They use a sequencing technology that we would sequencing approach that we refer to as sequencing by synthesis, which relies on reversible chain terminators. And remember when we went back to Sanger sequencing, how we had these terminating dideoxynucleotides? It's the same kind of concept in this case, right? The same type of idea. Here we have these, each of these terminators, right, associated with what we would consider to be a different base, right? Blue, green, yellow, and red. So each of them associated with historically what we would think of as the DNA base. And how it does this is it does something that we refer to as bridge amplification. But it's a pretty straightforward process. You take a DNA fragment, just like we had before, right? We attach adapters on either end, either through ligation or amplification, just like we did before. So same thing as 454. But the difference is that once we have these adapters, instead of putting them onto a bead and then putting them into a plate, now we have a slide with a whole bunch of little spots, primer adapters spotted onto that plate, which allows all of these samples to bind, all these blue spots to bind to these samples. And then what happens is it undergoes something called bridge amplification, and it'll generate these clusters. And within each of those clusters, right, we'll have identical sequences. And then once we have those clusters established on that plate, we'll then start adding in our, using the reversible terminator chemistry to do our actual sequencing. And how it looks is basically you have these cameras that look at each of these spots, and they know that each of these spots represents one cluster, and they'll just track the sequences, the colors that are coming off of that cluster in order. So every cycle, they'll count, what is the color that I got off of there in that one spot. And based upon that progression of flashing colored lights, again, similar to 454, but now instead of just one color, we've got 4 colors, they'll do their base calling. And let's watch a quick little demonstration on it.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
So again, you can attach these primers either through ligation or through amplification. There's multiple ways to go about doing that.
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with elan, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell.
So notice it's a slightly different process than the other example where every time you brought in the template and it attached to the bead and then it would come off and it would reattach and it would come off and it would reattach. In this case, it makes one copy, an original copy that attaches to this primer attached to the plate, and then it makes additional copies of that original copy with bridge amplification. And I think this is one of the reasons that we also see, I think, a less of an issue with, among other reasons, with error rates on the Illumina sequencer.
Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted.
Note that in this case, you're not flowing each. You're not cycling through A's, then T's, then C's, then G's. You're actually washing with a mix of all four at each cycle. So you don't have to wait, and it actually takes less time to sequence, right? It's a little more streamlined in terms of that process.
This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process.
So that what they just rotate around. That's the slide that you actually put into the machine. So it's just a glass slide.
Image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer.
So that was a bunch of different steps to basically help ensure that the system is looking at the same sequence, right? So it did a sequence at one end of the sequence, then it does the bridge so it can sequence at the other end of the sequence. And in this way, we're able to generate something in Illumina sequencing that we refer to as paired reads, right? Reads that are a pair of reads that are from the same sequence, right? One from each end of that sequence. So here they're talking about first we sequenced read 1, then we sequenced read 2.
As with read 1, the sequencing steps are repeated until the desired read length is achieved. The read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation.
So in this case, it's referring to the fact that we can add different adapter sets or barcodes, so a specific sequence that we know is we can associate with a specific sample during the library preparation step. So every time we take a sample and we build it for sequencing, build it into a library, we can add a barcode to it to make sure that we know that it belongs to that specific sample.
For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification.
So this is actually, what they're talking about here is just one application of how we might use the sequencing data. So Illumina sequencing and this process of sequencing by synthesis is incredibly powerful. It's the most widely used technology out there nowadays. We can see how it was built on a similar, and this is an old, this is like the GA2, one of the first sequencers that Illumina built. They now, we have a MySeq in the lab here at CSUMB, and it's a lot smaller than that, so we see a progression through time and what these things look like. It's an incredibly powerful technology. The crazy thing about this, just so you realize too, is historically these sequencing machines, it's been a little bit more stable in recent years, but during the late 2000s, early 2010s, this technology was evolving so rapidly that we were buying these hundred thousands of dollars of these machines that just would end up becoming doorstops after a period of time. It was kind of wild because the technology was improving so dramatically so quickly. One thing that's important to realize is what is the data look that you're getting off these Illumina reads, right? What is what makes it look? What does it look like when it comes off? And if we think about what we're sequencing, we're actually sequencing a read that looks like this, right? That has some sort of amplification element at either end, right? In these cases, there are these adapters, these primers, right? And then it can often have a barcode in it. And sometimes that barcode is located, there are different ways that people design these barcodes. It can be located just adjacent to your sequence. It can be located within the adapter. And there are even dual index barcodes where you have one in this end of the adapter and another one in this end of the adapter. And I think it's important just to realize this is that because we set these things all up the same, we always know that, when we have this first read, that it's going to be coming from this end of the read. And then when we eventually generate our read 2, notice that it's flipped over. So actually it's going to be the reverse complement of this end of the insert. Another thing to recognize is that we've added in all this extra amplification business, all these extra primers and barcodes. We want to make sure that those don't get sequenced by accident, because they definitely can be. So usually we're not that worried about trimming anything from the 5' end, because we know that sequencing will always start at a very reliable position. But you can sequence through this read and end up sequencing part of these adapter primers. Or maybe there's something similar within the sequence and you start sequencing in the middle of it so you get the rest of it. So that kind of stuff does happen. And so we do have to make sure that we think about any extra material that might have carried over from the sequencing process into our data, because it's just going to cause us problems later on. When we think about Illumina reads, probably one of the things that it's best understood for is introducing these paired end read processes, and also its sister, the mate pair data type. So paired end read means that basically when we're sequencing, right, we're sequencing two reads from the same sequence. We're basically sequencing the ends of those sequences. And so what's powerful of that is that we know that the two sequences that we generate come from the same piece of DNA, and we just have some unknown stuff in the middle, right? And so it's like we have one sequence where we have sequences on either end, but we don't know what's in the middle. We might know how much space is there. We might not. We might just have a good idea of it. But that actually affords us a lot of power later on down the line. And so, if we were going to take these sequences, we want to take this unknown fragment of DNA, we want to put it into our genome and figure out where it might go, right? In our genome, we're going to map it to our genome. It's helpful if we have two different spots to help anchor it rather than just one, right? That's incredibly helpful. And so one thing that you'll notice is that when we map it, like if it matches and the size, the distance between it is what we expect it to be, or within range of that, I'm like, oh, it's a good match. We can also get information from that, like if there's more space in there than we would otherwise predict or expect, we may think that there's an insertion in some part of the DNA where this is matched, right? Or we may think there's a deletion if there's less space than we might expect. If we see this sequence mapping, these two pairs of sequence mapping to multiple places in the DNA, in the genome, then we might identify those as being tandem duplications, right? Or if we see that the directionality of, say, like the second read suddenly changes and it's pointing in the wrong direction when we map it, that means that we could be looking at an inversion. Or if we have a sequence and we find that it maps perfectly in one position and then pieces of it map in other positions, we might think, well, this might be a repeat element insert since it's small. So like this one part of the gene, one part of the DNA sequence is repeated where this part is not, right? And so there's all these different possible outcomes and extra information but when we know that they're in pair, can really help us. And we're also going to be looking at some mate pair sequencing. Mate pair sequencing, so typically paired end sequencing is going to be, inserts ranging from no inserts, where actually the forward and the reverse reads overlap with each other, to maybe as large as like 600 base pairs or 800 base pairs. I mean, usually it's more along the scale of like 50 to 300 base pair inserts. That would be a more sort of expected distance, maybe 400 or 500. But not much larger than that typically. So mate pair sequencing, in this case, this is for when we want to get paired sequences but are longer apart, right? And so we might have a longer fragment, say instead of 1,000 base pairs or 500 base pairs or 300 base pairs, we've got something that's 25,000 base pairs, right? And what happens is we biotin label the ends of it, we circularize that molecule, right? And so we've got these biotin markers at this spot, right? We circularize it, we shear it, we pull down all these biotin pieces, right? We then ligate our adapters onto the end of that, and then we sequence the ends of this sequence, right? And since we know that this whole segment was about 25,000 base pairs, we know that if we think about all the space from here down to here, right, then that would be approximately the length of the original fragment, right? So if we think about the distance then that's between these two sequences, it's approximately the length of what was between these sequences originally. So if it was 25,000 base pairs, now we've got something that's probably like about 24 something, right, between these two reads. But also notice one thing: the paired end sequences you would have had it in this direction and in this direction. So on the forward strand and on the reverse strand. In paired end sequencing, it switches, right? So you've got the first read is actually the reverse complement and facing away from each other. And then, and then The forward read is at the other end, and again, facing away from each other. So something to remember as we go through this process. I think one important thing to note about Illumina sequencing is it is not perfect. It does have biases. For instance, in really biased GC content regions, so at the extremes of GC content, it tends to underrepresent those types of situations. So it is important to understand that this is not a perfect technology, but it is the most widely applied. And this sort of advent of what we referred to at the time as next generation sequencing, and you'll often refer to as NGS. I think that the take-home message is that massive parallelization was incredibly innovative in this process. It reduced the costs absolutely enormously and reduced the amount of effort exponentially. I mean, it's just been wild how much it's improved our ability to obtain sequencing data. So we talk about 1,000 monkeys on 1,000 typewriters, recreating Shakespeare's works. But here we're really talking about 100,000 to 100 million to billions of concurrent reads. I think the NovaSeq now puts out somewhere around 20 billion reads per run. So the amount of information that we're able to generate now is really, really impressive. So we have our sort of so we had our next generation sequencing. Now we get on this. I mean, I'm not really going to get into this like second generation, third generation, all this sort of stuff. But there has been a set of technology that has come after that, right? Pacific Biosciences is one of these approaches. They came out with something referred to as single molecule real-time sequencing, or smart sequencing. And let me show you a little bit of a demo on how that works.
Pacific Biosciences is developing a transformative DNA sequencing technology that will revolutionize the field of genetic analysis by enabling researchers to answer questions important to human healthcare. We call it SMART, Single Molecule Real-Time DNA Sequencing, a breakthrough technology based on the natural process that occurs every time living cells divide. Prior to division, DNA is replicated by enzymes called DNA polymerases, which efficiently duplicate entire genomes in minutes by reading the DNA and sequentially building a complementary strand with matching building blocks called nucleotides. Pacific Biosciences smart sequencing harnesses the power of the polymerase as a sequencing engine by eavesdropping on it while it works to replicate DNA. This approach is enabled by two proprietary technologies. The first is phospholinked nucleotides. To visualize polymerase activity, a different colored fluorescent label is attached to each of the four nucleotides, A, C, G, and T.
Sound familiar, right? So definitely a different approach here using this DNA polymerase to sort of drive the sequencing process. But again, you have these four different colors. You have this light sensitive process, right? So some common things. We also have a slide that holds beads.
In contrast to other sequencing approaches, our phospholinked nucleotides carry their fluorescent label on the terminal phosphate rather than the base. Through this innovation, the enzyme cleaves away the fluorescent label as part of the incorporation process, leaving behind a completely natural strand of DNA. This enables us to exploit the inherent properties of the DNA polymerase, including high speed, long read length, and high fidelity. The second key technology is a nanophotonic visualization chamber called the Zero Mode Waveguide, or ZMW. It enables observation of individual molecules against the required background of labeled nucleotides, while maintaining high signal-to-noise. The ZMW is a cylindrical metallic chamber, approximately 70 nanometers wide, that is illuminated through its glass support, creating an extremely small detection volume, just 20 zeptoliters.
So again, in this case, not a bead per se, but still this sort of slide with these wells approach.
Nucleotides diffuse in and out of the ZMW in microseconds. When the polymerase encounters the correct nucleotide, it takes several milliseconds to incorporate it, during which time its fluorescent label is excited, emitting light that's captured by a sensitive detector. After incorporation, the label is clipped off and diffuses away. The whole process repeats, creating sequential bursts of light corresponding to the different nucleotides. These are recorded, thus building the DNA sequence. Our smart sequencing harnesses the polymerase's natural ability to synthesize 10 or more bases per second over thousands of continuous incorporations, leading to high speed and long read sequencing.
It's like a sequencing
Additionally,
rave.
our smart technology design allows simultaneous multiplexing of thousands of ZMWs in parallel, all concurrently replicating DNA in real time. The data is assembled into whole genomes and analyzed by researchers. Our smart sequencing technology will ultimately translate nature's ability to replicate an entire genome in under an hour to an instrument that will sequence a human genome in minutes for under $100. This unprecedented level of performance will completely redefine the field of genetic.
So, yeah, very powerful technology, and for the vertebrate genome project is now like the dominant technology. It's actually improved even since then. I won't go into the details of some of the other ways that they've adapted this. That's kind of an older video, but definitely one of the becoming incredibly reliable from accuracy and precision standpoint, and just a really useful technology. A little bit more expensive, a little bit harder to generate as much as you would for Illumina sequencing, but still a very useful technology. Another incredibly important process has been the advent of some chromosome conformation capture methods. And these are methods we refer to as high-C methods. And high-C is a method by which we take advantage of the fact that along chromosomes, we actually, in chromatin, there are cases where we end up having these loops where we might have a large section of DNA, say hundreds of thousands of base pairs, or tens of thousands, or 100,000, or whatever, long loop, and it's connected here in the middle by some sort of, nucleosome structure or something of that case, or something of that type, right? And basically, so we have two sections, and we want to think about that mate pair example, right, where we had two reads at the end of a long loop, right? In this case, they're already connected. We don't have to take that biotin piece. They're already connected here. So we've already got these reads that we know are this far apart. So however far that is, right? We can then identify these reads that are approximately that far apart. And what we essentially do is we cross-link, using paraformaldehyde or some sort of cross-linking agent such as that, so that we end up with having the two strands connected to each other in this spot. We then break off the ends, right? We ligate these two ends together, and we end up with a read where one end and the other end are approximately this far apart from each other. So that could be hundreds of kilobases away from each other. So hundreds of thousands of base pairs away from each other. And so it's become a really great method for getting sequences, paired sequences. So we know these sequences are on the same chromosome, right? But they're really far away from each other.
Proximo High-C.
So there are two companies that do this. I'm going to briefly show you a video from Proximo High-C, but Dovetail Genomics is the other group. Dovetail is just up the road in Santa Cruz. Excellent company as well. Both of them provide this same sort of approach of generating high-C data.
We start with intact tissue from plants, animals, and human samples or cultured cells. Chromatin is packaged into three-dimensional structures that retain a relationship between genomic and physical distance. Sequences that are closer on the same chromosome are also closer in physical space. Our method exploits this relationship between linkage and proximity to enable whole chromosome scaffolding and phasing of genomes. The DNA in the sample is cross-linked in vivo to fix DNA sequences present inside the same cell. Cross-linking traps sequence interactions across the entire genome and between different chromosomes. Cross-linked DNA is fragmented with endonucleases. Fragmented loci are then biotinylated and ligated, creating chimeric junctions between adjacent sequences. This process is called proximity ligation. The more often two sequences are joined together, the closer these two sequences are in genomic space. Biotinylated junctions are purified and subjected to paired-end sequencing. The proximity ligation reads are then mapped onto a draft assembly. Proximity information is used to assign contigs to chromosomes and order and orient them along chromosome scale scaffolds. This results in fully scaffolded chromosomes of virtually any size. This process also detects structural variation and corrects assembly misjoins, as well as maps the three-dimensional conformation of chromatin within a population of cells. Bring the power of Hi-C into your lab today. Learn more by visiting Phase Genome.
So it's really this technology, I think, which has been one of the principal steps for us allowing to get to the point where we have chromosome level sequences. And so it's been really, the advent of longer sequences and then these Hi-C methods have been so important to us moving forward. The last of these sort of sets of methods is the Bionano system, which is the iris system. This is a really interesting system because it's actually not necessarily generating a sequence how we would traditionally think about a sequence. And so what happens is you have high molecular weight DNA, in other words, DNA that's really in long pieces. You label specific sequences across this piece of DNA, and then you load it into the system, and it's going to generate these images of the ordering of these labels, right? And by using that ordering, we can then take our genome assemblies and order the different fragments that we've assembled, these different contigs, shorter pieces, short thousands of base pairs long, hundreds of thousands of base pairs long, order them into one contiguous piece. So we can kind of figure out how they would work along a chromosome. So as an example of this, let me show you how this system works.
This is Sapphire from Bionano Genomics. With it, you can directly image the genome at high resolution to detect variations such as deletions, duplications, translocations, and inversions, known as structural variations, with sensitivity as high as 99%. The process is simple and automated to enable routine use in disease research. Structural variation commonly occurs in a broad range of diseases, including neurodegenerative diseases, intellectual disabilities, and various types of cancer. In fact, the first targeted cancer therapies were based on gene fusions, a type of structural variant. Given the role structural variants play in both understanding and developing potential targeted therapies, it is important that structural variants are accurately identified or called. To do this, there are two fundamentally different yet complementary approaches: sequencing and whole genome imaging. In short-read sequencing or next-generation sequencing, DNA is read in short segments of about 150 base pairs, and the read sequences are compared to a reference genome. It can characterize small variants, but structural variants are often tens of thousands of base pairs or longer, so short-read sequencing is not an accurate way of calling them. What's more, 2/3 of the genome is repetitive, and structural variants in repeat regions are often invisible by whole genome sequencing. Whole genome imaging with the Sapphire system takes an entirely different approach to understanding the structure of DNA through imaging. It has proven to accurately identify structural variants ranging from 500 base pairs to whole chromosome lengths with sensitivity as high as 99%. First, native ultra-high molecular weight DNA, multi-megabase pairs in length, is isolated. Fluorescent labels are enzymatically attached to these ultra-long molecules of DNA at a specific sequence motif that occurs throughout the genome. The resulting label patterns are unique for each section of the genome. The full length of the DNA remains intact during isolation and labeling, preserving the architecture for the next step, imaging the genome. The labeled DNA is pipetted into the Sapphire chip and placed in the Sapphire instrument. In the chip, the DNA is uncoiled or linearized across thousands of parallel nanochannels and imaged, revealing its fluorescent label patterns. By capturing these images in repeated cycles across the hundreds of thousands of nanochannels on the chip, all the images necessary to assemble a map of an entire genome are captured.
So notice that instead of assembling a sequence, in this case, they're assembling a progression or a map of these fluorescent markers. So it's a different method, but it's the same conceptual idea of assembly.
Algorithms extract molecules from the images, and then Bionano algorithms construct consensus genome maps. Bionano Access software also allows easy visualization and filtering of SV calls using our built-in control SV database. From the thousands of SVs found in any human sample, you can remove variants present in controls and narrow down your SVs of interest to a handful with just a few clicks. This is all enabled due to the extremely long molecules isolated and imaged with the Sapphire system. Want to use whole genome imaging?
So you can see that there are, again, there are some parallels to other processes, but a very different approach to doing this, but used for specific purpose, especially when thinking about how we might utilize what is a traditional area of inquiry, genome maps, to help aid with some other problems that a traditional sequencing approach might not address. So lastly, I'll talk about what I put future comma because we might already be in the future. We could call this third generation. I don't know if we call the last one third. I mean, I don't really know what the point is, but there's a whole other area that kind of is. It's very similar to the PacBio idea of single molecule sequencing, but I would say that Oxford Nanopore sequencing kind of is its own animal, and that's because technologically it's very different, right? So Oxford Nanopore works in this way in which it uses a nanopore, and a lot of these nanopores actually have their original construction and innovation was developed up at UCSC, just up the road. But it uses these nanopores to take a single DNA molecule, thread it through this nanopore and this membrane in order to track the flow of ions that these pores create and record the electrical signal that's associated with this DNA as it goes through this nanopore. So let me show you. They do a good job of explaining it on this video.
For DNA sequencing, intact DNA strands are processed by the nanopores and can be analyzed in real time. The nanopore sequences the fragments that are presented to it regardless of their length, rather than generating reads of a specific length. This could be reads of hundreds of kilobases or more. Nanopore long reads simplify assembly and sequencing of repetitive regions, also improving the speed of species identification in metagenomic experiments. So what happens during nanopore sequencing? The DNA strands to be sequenced are mixed with copies of a processive enzyme, shown here in green. As the DNA enzyme complexes approach the nanopore, the single-stranded DNA is pulled through the aperture. The enzyme ratchets the DNA strand through the nanopore one base at a time. The speed of the enzyme can be controlled. More data is yielded per second the faster the enzyme runs, but with no deterioration in accuracy. As the DNA moves through the pore, the combination of nucleotides in the strand being processed creates a characteristic disruption in the electrical current. This nanopore signal can be used to determine the order of bases on that DNA strand.
So, it's detecting an electrical current. Similar to anything we've seen before? Well, it's a little bit similar to, if we want to think about the ion-torrent idea of tracking pH, right? Different, very different approach, right, than a lot of the other methods that we've seen, right? Not using the color system that we've identified before, and not trying to actually use these, these nucleotide-based chemistries approaches, but still we see some of the same ideas, a few of the same ideas floating through. So we can still see some connections to some other ideas.
Nanopores have processed read lengths of hundreds of kilobases, and when a nanopore has processed a complete read, it will start a new one. Nanopores start to stream data as soon as the experiment begins, with base calling taking place locally. Users can also take advantage of epitome workflows for real-time analysis, continuing the experiment until sufficient data has been analyzed to determine the answer to a biological question. With nanopore sequencing, the user has the ability to run the experiment until the answer is reached, rather than working to an arbitrary instrument runtime. The workflow described here is adaptable and is used to sequence a variety of molecules in real time, including genomic DNA, amplified genomic DNA, and PCR amplicons. In the same way that native DNA can be analyzed, RNA can be sequenced in its native form or by using cDNA.
So in this case, when it talks about RNA, it means that you may be able to actually sequence RNA directly. One of the really compelling things about this technology as well is how innovative it is from a portability perspective and size perspective. So in addition to sort of your traditional larger sequencing effort, like the PromethION, which can put through like an enormous amount of DNA sequencing in a period of time, it is a little more expensive to run the sequencing, by the way, per kilobase, per megabase. But you also have like these small little MinION sequencers, and these things, like this little MinION right here, that's basically it fits in the palm of my hand. So that's how small the sequencer has gotten. It's pretty wild how this stuff works. So there's just that portability now leaves us with the option of taking sequencers wherever we want to go. I had to do sequencing at my house when we were first in COVID shutdown, and that was totally feasible because that's how portable this technology is. So there's all these ways where sequencing is just changing dramatically. Maybe soon we'll be able to just they're trying to develop like a cell phone platform where you can just do it right, plug it into your cell phone. So all kinds of really interesting advancements and things happening in the world of sequencing and ongoing. So some final thoughts here. One, overall, just DNA sequencing has become vastly faster and more affordable over time. Generating data is no longer the bottleneck. Understanding it is, hence the reason for this class. So bioinformatics is in high demand, and each technology has its own idiosyncrasies, advantages, and disadvantages, and it's important to you as a bioinformatician to understand what those are. I forgot to mention, Oxford Nanopore, it's using those electrical signals. It's not looking at each of those bases individually. So because it's looking at that overall signal, it also sometimes has suffered from some issues with homopolymers, right? So there are some things we want to be careful of when we think of these, but it's a really exciting time to deal with this kind of data, this kind of innovation. And I think it's really cool to see the history of the evolution of these technologies, because we see the points where they're connected. We see the innovations that people have been able to come up with to change them and to create an entirely new technology or a different technology, a better technology. And I'm just excited to see where this stuff goes, because I think it's going to be really, really interesting in the future.